Thank you.
Okay, I'd like to thank the organizers for inviting me to talk. And today I'm going to tell you a little bit about one of our main undergraduate projects that we developed over the pandemic, and also a little bit about what I did sort of the first summer of the pandemic to give you an idea about one of our other things. So basically, I'm actually from a wet lab department. So all of our undergraduate projects are usually things like pipetting or working with fruit flies or something like that. But with the pandemic, everything shut down. And we were basically told, well, I guess undergrads can't do projects. And I said, wait a minute, I can come up with something. I'll figure out something. Because it's very important for undergraduates to have projects. Sure, they get the research experience, but they also get the mentoring, the training, and other preparation for graduate school and professional school. So it's really important that our undergraduates could get some experience before they graduated. Okay. So in our lab, we have a database of proteins that are called moonlighting proteins. They have more than one function, physiologically relevant functions shown in publications through biochemical and biophysical studies. So I said, okay, let me take a few students, and that first summer, I'll just come up with something that they can do. And it turns out I set up something where they could do some background reading on moonlighting proteins and other proteins. And we started with some annotation, adding in more proteins to our database. So they learned about using UniProt, using BLAST for sequence alignments, understanding concepts about protein structure and function and sequence and all these different things. And then as we got quite a few more proteins added to our database, I also developed a second phase where our group grew, so I could have little subgroups, and they could learn about using online tools to analyze some of the sequences and structures. So in the bottom here, I have just a little example of students taking all 500 sequences and using an online server and learning about how many of them are predicted to be transmembrane proteins. So this is something, again, they could learn about actually having a hypothesis and looking at some data, having a very clear output from programs like this, and then saying something about how many proteins are transmembrane proteins. And they also learned about things like prediction methods and what it's based on, the biophysics and all that. Okay, so for that first summer where I was originally going to do this with 3 students, word got out. I added in a few students from a summer research opportunity program. Other students contacted me from other schools, and also high school teachers and some high school students. So we had quite a good group. It grew quite a bit. We expanded not only the number of people involved, the number of proteins in our database, but also the types of information in our database. So I had some grad students actually come back and volunteer and help us really improve our tool. And we added about 200 proteins and more information about all 500 proteins during that summer. We provided some research opportunities online for students. We actually, all the students, high school teachers, everyone involved got to be authors on our publication in nucleic acids research. Students were able to complete the requirements. If they were a summer student, they wrote a report at the end. Some students continued into the fall and got course credit, honors thesis, and other things. So there's more information about that project in a paper in the Biophysicist, and also I have a webinar with the Biophysical Society that talks more about that project. Now that we saw that could work out, and the pandemic was continuing into the second summer, but now we had a little more warning. So I was able to apply for funding from our National Science Foundation, and this is through they have research experiences for undergraduates, usually in person, but I got a special grant 1 year for something because of the pandemic. Okay, so this is what's a rapid REU. So we had 7 student participants, 7 faculty mentors, and one benefit of this being online is I could select wonderful faculty members doing current, exciting research from different universities who are very enthusiastic about taking their time out of the summer to mentor students as a volunteer. Another key person was our near peer mentor. It's a graduate student of mine. She's somewhat closer in age to the students than I am. so they could feel much more comfortable coming to her with concerns and just talking with her and everything. And she helped out in a number of other ways as well. So this is just a brief outline of our schedule of activities. Before we started, we did have a mentor orientation to talk with the mentors about working with students. These were often students from underrepresented groups who maybe had very little or no experience in any sort of research before. Then the first few days of our actual summer program, we had a participant orientation. So we went over, of course, the schedule, the plan for the summer, background information on protein structure and function. Very important. Again, they were coming from a whole variety of different previous experience and training as undergraduates. And introduction to research. What do we mean by a hypothesis? How do we come up with a hypothesis? How to keep a notebook? Even though these are, online projects we're doing, we still have a hypothesis. There's still background meeting. There's still methods that they should write down, observations, conclusions, maybe having to go back and repeat something. The main part of their 8 weeks was a mentored research project, and I'll talk a little bit more about that afterwards. But we also came together, the 7 participants, me and my near-peer mentor, for about twice a week workshops and presentations. So these covered things like applying to grad school and paying for grad school, because you might need to get fellowships depending on what type of school you go to. And we had some grad students do a panel discussion, answering any questions they had. We had some discussions about careers in biotech as well as academics. And quite a few workshops on skills, how to read a scientific paper, write an abstract, give a talk, make a poster, all those sorts of things, as well as discussions about diversity, ethics, creating an individual development plan. And my near-peer mentor was awesome. She helped out with some of the preparation of these workshops, but she also created a bunch of icebreakers to help get the participants to really be participating in the workshops and not sort of sitting back and listening to me talk or guest speaker talk or something. So really getting everyone involved. And she also had experience in a gap year, which is a year or two between college and grad school. So she could talk about how you arrange something like that. She worked in a biotech company for a while. She also had these weekly check-ins. So she met with each student each week individually to find out if there were concerns, questions, anything that we could help with, ways we can improve the program, anything like that. The participants had some assignments, so they needed to write an abstract, a poster, a paper, and a talk for our symposium at the end. So we had an online symposium. Each student gave a brief talk, and the mentors were invited and members of the labs. And I created an abstract book, and I printed all their posters and printed certificates, mailed all that off to them so that they could present they would have a sort of a memento and information that they participated in everything, but they could also give their posters at their university or at other meetings, and some students did follow up and do that. And then we had a number of evaluations. During the 8 weeks, we had a few evaluations where students gave evaluations of the program, but also some information about how they were progressing. Did they have any sort of figures ready for their papers, for their talks? Are they understanding the material, suggestions for the program as well? And we had the professors evaluate them every few weeks as well, so we could make sure that they were showing up and doing the work, and again, helping us think about if we recruit students for another program. These are just examples of some of the research projects students could do online. Again, we're coming from more like the wet lab backgrounds, the structural biology backgrounds and everything, but the professors were really wonderful in carving out the type of a project that a person new to research could do in about a 7 or 8 week period. So molecular dynamics simulations of conformational changes, computational docking of ligands to proteins, mapping of ligand binding sites, computational studies of allosteric pathways, and other types of computations like liquid-liquid phase separation. And if I have a couple minutes, I'll just tell you briefly what one of the students did with me, and then I continued into the fall of the pandemic with other students at my university too. So we had this whole group of proteins that were found from a number of labs from proteomic studies that their enzymes and pathways like glycolysis, citric acid cycle, carbon, nitrogen metabolism, and guess what? They all bind RNA. How, why, we don't know, but that's what we're interested in my lab. So we had the undergrads help us take whatever information we could get about these proteins and any cross-linked fragments of RNA, fragments of protein from these studies, and think about how is a protein doing these two things. So we have a lot of information about these proteins from many decades of studies about their folds and sequence homology and constellations of active sites or constellation of amino acids in the active sites that are involving catalysis. But we don't necessarily know much about how are they now interacting with RNA, but we had bits and pieces of information. So the students could help out in mapping on the three-dimensional structures, the active site amino acids, these fragments of proteins that were found to actually be binding to the cross-linking to the RNA, post-translation modifications, conserved regions, other features of the protein, and they could learn to do things like multi-sequence alignments with BLAST, using protein databases like UniProt, sequence conservation with Conserve, and molecular modeling rendering with PyMol. And this is just a simple example of that. This is isocitrate dehydrogenase. It's a dimer, so we have a blue subunit and a green subunit, and we have a crystal structure, with a cofactor bound in the active site. So the students could render the structure, look at it, move it around, think about what its structure's like, and they could highlight that cofactor in the active site in dark blue, and then highlight the fragment of the protein that was found in proteomic studies to bind to RNA in yellow. And then they could look at this result in 3D, move it all around and everything, and it could help us come up with some hypotheses, rather straightforward in this case, but we have this peptide binding to RNA that is basically right in and around the active site. So we could say, okay, does RNA binding affect catalysis? And this is something we could then follow up in when we were able to go back into lab the next semester. Okay, so overall the project went really well, and we are planning to do another REU this time. The full 3 years that NSF funds would be 10 students, 10 professors, etc. I'm sorry, did I make that too short? Okay, so in summary, even though I'm coming from a wet lab department, we were able to create, with relatively short notice, some online remote student research projects. And we had some great outcomes. Students were able to learn how to make abstracts and talks and posters. Some students who continued got course credit. We had honors thesis, some posters were presented at meetings. There were actually a couple talks given. My near-peer mentor was invited to give a talk at one school and things like that. And they actually did, in that short amount of time, help us to move some of our projects along a bit, helped us to pick out from this whole group of proteins, some proteins we wanted to work on specifically in the wet lab afterwards. Now, I think that even without a pandemic, these online research experiences are really important. We have a lot of students who are at schools that don't have large research programs, that might be very, very hard for them to find a research project at their own university. And a lot of students can't just go to one of these usual REUs that NSF funds. It takes 10 weeks out of the summer. A lot of students can't just kind of leave for 10 weeks. Some might have responsibilities at home with younger siblings, for example. And some students have physical challenges. Again, they might not really be feasible for them to travel for 10 weeks to some strange city and work at a lab bench, pipetting all the time and everything. And I will note, we also have one person volunteering with the database who's from Ukraine, so we could provide an opportunity to students who maybe, again, might not have a lot of research opportunities where they're located, maybe can't really travel, and she can work on her own pace as things are going on in her country. So I'd be happy to share more information with you today. I have a poster, C38. Also, there's a lot more information with some supplementary material to tell you a little bit more about workshops and other things that we did, and a lot of references that we used to help us build up some of this material in the Bioinformatics Advances. Bioinformatics Advances should be coming out any day. It's in press now. And I'd like to thank my near-peer mentor. She was great, helped out in many, many ways, and all of these students and high school teachers and others who helped create the database over the years, and especially came in during the pandemic, help us build that database, and also worked on these different projects and everything. NSF funded that the summer program during the second summer. And also I'd like to recognize also that a lot of this, the database and some of the modeling and everything, was based on a lot of publications from a lot of labs. I wasn't able to really reference them all, but we do have quite a few of them in our papers as well. And I'd like to thank all of you for all of your time and attention.
Thank you very much, Connie. Have we got any questions for Connie? Russell.
So I was wondering if you could elaborate on the last point of going forward and taking advantage of what you've developed now that we're sort of post-pandemic. Would you see this continuing as a purely online program, or doing something more hybrid with an in-person and an online component?
Yeah. So for a number of reasons, I do like this opportunity for students online. And I think there are a few other little perks that I think are great too, is that in finding mentors, one-on-one mentors, a lot of people's summers are very complicated. And this is a way that mentors could also participate, and as well as grad students and everything. I have been considering the possibility of doing something that's a little bit hybrid, in that we could perhaps everyone, or at least all the participants, maybe not all the mentors because they might have a little more complex travel plans, but like for a few days of orientation at the beginning. So maybe we all meet in Chicago and have some of the orientation and get to know each other a little bit, so that when they do come together in workshops and everything, they know each other already. They don't have to worry about feeling uncomfortable. So that was our plan for this small group, but certainly this could be done in a number of different ways. We have a very large number of undergraduates at my university. We have 1,600 biology majors. So there could certainly be an aspect where we did some things in person and some things more remotely. But for this specific summer program, the REU, I'm planning either fully remote or potentially a starting orientation that would be in person.
Thank you.
So I just have one more question at the back.
This is not a question. This was a great talk. I just wanted to make you aware that UniProt, you're using, apparently you're teaching the students UniProt and looking at the literature and the proteins. We have a community submission page where if you have literature that is not in UniProt, you can add it and be acknowledged if you use ORCID with ORCID. And so the students could have an extra thing if they, in the project, in addition to doing all the annotations in the Moonlight database, you can actually tell them, okay, you can add this literature in UniProt with some maybe some annotation, like the function that they found in the paper. And then they can cite that they contributed to the database.
Oh, thank you. That's wonderful. Yes.
We can talk afterwards.
Yeah.
If you want.
We also, before that, we did have about 300 proteins in our database. And so we are linked with UniProt, but I didn't realize about the ORCID number and everything.
So the students could actually add, you could add, I mean, they can do it anytime, but since they're doing that project, and they could add that literature and annotations, and they will be visible with the.
Okay, great. Thank you.